Good morning, my girls. So let's go ahead and talk about meiosis. So as you should know from the videos that you guys have watched, meiosis is the type of cell division that is used to produce our gametes. Gametes is the fancy name for our sex cells, our sperm and our egg cells. In females, we have eggs, and in males, we have sperm. So in this PowerPoint, I'm going to go over some basics and kind of get us settled with some terms that you probably heard in the video, and you've also heard from the mitosis videos, but I'm kind of going to just go over those a little bit more to make sure that we are all on the exact same page. Okay, so the first thing I want to say before I start talking about everything that's going on this slide is the main difference between mitosis and meiosis is that in mitosis, the goal of mitosis is to make two genetically identical daughter cells. You want them to be exactly the same, because that's the process that you use to make more of you, or for whatever organism to make more of itself. Now, for meiosis, the goal is to make not two, but 4 genetically different cells. Okay, so we're making 4 that are genetically different that only have half the amount of chromosomes. They're only going to have 23 in humans instead of 46. And remember, this is because if there were 23 chromosomes in the egg and 23 chromosomes in the sperm, when that comes together, that makes 46. That's a full human, ready to go. If you had 46 chromosomes in a sperm and you had 46 chromosomes in an egg, when that came together, that would be 92 chromosomes. That's entirely too many, and that fertilized egg would not survive, if it even was able to be fertilized with that number of chromosomes. So again, meiosis is making 4 genetically different daughter cells that each have half the amount of chromosomes. If they had the full amount, that would be no bueno. So let's talk about homologous pairs. What does that word mean? What does that look like? Well, I'm about to show you. So in a typical somatic cell or a body cell of a human being, there's 46 chromosomes. I think we're all pretty good with that fact. If we were to look at human chromosomes during metaphase of mitosis, which we can do, that's kind of like what you did in the virtual cell lab for mitosis, except you were looking at onion cells, not human cells. Just a disclaimer there, that was onions. But if you were to look at human chromosomes during metaphase of mitosis, and they're all lined up on the equator, or the middle of the cell, the metaphase plate, you would see 23 sets of duplicated chromosomes for a total of 46 chromosomes, right? 23 pairs, 46 total. If we use special dyes, which we can do, and we do, in fact do, if we used special dyes and we looked at the pairs, we would see that they have matching patterns. So what does that mean? If you look on the right, okay, in this picture over here with the pink and yellow and green chromosomes, if you look, you can see that they both look very identical. They each have the hot pink at the top, they each have the yellow next, then the green, then the blue, then the yellow again, right? So they are exactly the same. They have these matching patterns. Now on the next slide we're going to talk about what makes them different from each other, but for right now you need to notice that they have these matching patterns. Remember that each chromosome is going to be duplicated prior to division, because you have to make a copy, you have to replicate, right? That's S phase. That still happens for meiosis, for cells that are going to go through meiosis. They still have to make a copy of their DNA. Otherwise, the cells at the end would have 1/4 of the DNA instead of half of the DNA. And so they're going to duplicate, and what you'll see here is if you look over here This one is showing you what a chromosome looks like once it's been duplicated, right? So this guy that I'm going to circle on this side, that's one of the sister chromatids of this chromosome. And this guy right here is its exact copy. Okay, that's its exact copy. That's the other sister chromatid, and they're attached in the middle by the centromere, sometimes also called the kinetochore. Almost every single chromosome in the human genome is going to have a twin that resembles it in length and in its centromere position. So what does that mean? That's like this guy right here. This whole guy, right, perfectly matches this friend right here, right? So that's its twin. Oh, Maggie, why are you whining? The matching pairs we call homologous chromosomes. So this whole entire picture together is looking at homologous chromosomes. Remember, homo means same, so it's a pair of the same chromosomes, essentially. And I'll explain why that's important in a little bit. So the two homologous chromosomes, they're both going to carry genes that control the same inherited characteristics. And that's kind of hinting at genetics, which I'm hoping we can squeeze in just a little bit before the end of school, because honestly, I did not realize how few class periods we actually have left to cover things, but I really want to get you guys at least a little bit of Punnett squares in there, because I know you guys have been looking forward to that for so long, and so have I. So I'm going to try to squeeze it in there. But the point is that the homologous pairs are both going to carry genes that control the same characteristics. So that could be things like eye color, hair color, how tall a person's going to be, how short a person's going to be, that kind of stuff. So genes are going to be located at a specific point on the chromosome called a locus. The plural of that is a loci. That root should remind you of another word. That word is location. So genes are just located at a certain place, and we call that a locus. So, for example, if you have a gene on a chromosome that codes for a person having freckles, that's going to be at a certain point, at a certain locus on one of the particular chromosomes. And then the homologous chromosome, or the guy that's going to match up with that chromosome, is going to have the same gene at the same place. So if we go back to our picture over here, let's say this guy that's pointing to locus, we'll just use that as an example. This yellow guy right here is going to code for freckles, and this one over here is also going to code for whether you're going to have freckles or not. Now, the difference comes in the different versions of these genes, and we call those different versions of genes alleles. And we'll talk a lot about alleles when we do genetics. But those two alleles may say something different. What do you mean? You just told me they were both coding for freckles. Right, but this one that you inherited from your mom might say, you are definitely going to have freckles. For sure, no doubt about it. But this guy that you inherited from your dad, he might say, no freckles. Nope. So we're still coding for whether or not we're freckles. It's still the same gene at the same location, but one version of the gene, one of the alleles, says you're going to have freckles, and one of the versions of the gene, one of the alleles, says that you are not going to have freckles. Okay, so that's what's different between the homologous pairs. They still have the same genes at the same locations. They just might have different information in those genes. Or another example might be that this one, let's maybe use this blue one down here. Maybe this blue one says that you're going to have blue eyes. And this one over here says that you're going to have brown eyes. Right? That's still the same gene at the same location, at the same loci. They just have different information in there. One says blue eyes, one says brown eyes. So hopefully that makes a little bit of sense. And I think that's everything that I wanted to say on this slide. And I'm actually going to stop this recording here and start a new one because I don't want to try to rush the next couple slides into the next 5 minutes.